Everybody knows that the availability of lots and lots of genomes is changing the way that we have to think about our research in microbiology. And one of the ways to do that, of course, is to look at what people have done in human genomics, which tends to be ahead of us, traditionally anyway. And in human genetics, genomics, there's been this big revolution in genome-wide association studies. So I'm going to be talking not so much about details of CLIME and things like that, but actually one of the kind of exciting new research areas that's facilitated by the kind of computational power that CLIME now gives us. So, provocative title, what's that all about? GWAS, religion or promising approach? Well, thanks to Mark Palin, who gave me this slide. Apparently, Barack Obama has now announced that GWAS is an official religion in the USA. So goodness knows what that means, but I guess the way it's a nice sort of framework for thinking about this talk. And I suppose you'll see, I hope, as I go through how I'm trying to use that to sort of describe what we do. But I guess what it means is we find lots of these associated SNPs or genetic elements that we can link to human disease or human geneticists do. But that requires this, this word association is not quite the same as cause. And if we think these SNPs, like the cancer gene or the schizophrenia gene or these various genes that are being discovered and written about in Nature all the time, if we think they're associated, that's one thing, but cause is different. And I think it's quite a nice title because really it requires faith. And I hope to show you that in some of our bacterial GWAS, we remove that requirement, or getting close to removing the requirement for faith, and we look for proof. Okay? Not that I'm making any particular statement about religion, I think whatever. It's just a context for making my talk slightly more interesting to a general audience. Okay, so in my lab, we look at genomics in natural bacterial populations. Many of you will know that most of the big problematic pathogens that we encounter exist principally as commensal organisms, so that are harmless. Okay, so if we think about things like Staphylococci, E. coli, and Campylobacter, three of the big ones, in UK, US hospitals. They're all basically commensal. Staphylococci on our skin, nasal pharynx, found in lots of animals. E. coli and Campylobacter found in lots of animal guts, gastrointestinal tract, and in the environment. But under certain circumstances, all of these organisms cause serious healthcare problems, okay, serious diseases. Obviously, MRSA, nosocomial infections, is the big famous Staphylococci one, and Campy and Campylobacter and E. coli, famous for gastroenteritis and other things. Okay, so I've showed this slide loads of times. Many people in this room will have seen me actually say this before, but I think it's quite a useful way of thinking about it. Traditional microbiology, the lab that I grew up in, as a microbiologist, we would typically start with a gene of unknown function in a reference laboratory strain, and we would think about, okay, what does this gene do? How does this change the phenotype of our organism? And the way you might test that often would be you'd make a deletion mutant and check how that changed the function of your bacterium. Okay, so you could think of that as bottom up. You start with a gene and then you're thinking about function. But obviously, more and more genomes, more and more genes of unknown function. What do we do about that? We can't generate TRADIS libraries for all of the bacteria that we isolate. It's just too much. So we looked at human genomics, as I've said, and we can think about top-down approaches. So that is where you take your bacteria and you group them by their phenotypic difference and then look for the genetic elements that are associated with one pool compared to the other, okay? It's an association study. Right, so Campylobacter is one of my favorite organisms in terms of my research. It's not very nice. It causes serious gastroenteritis. It's very common in the UK. Very expensive to the NHS, and it's a zoonosis, okay? And one of the key features, as you all know, if you're a zoonotic bacterium, one of the the ways that it's so widely transmitted is if it's found in lots of hosts and it enters the food chain, in the case of Campylobacter, through lots of routes, potentially. So one of the questions we had early on with Campylobacter when we started thinking about GWAS was, what are the genetic elements within the genomes of this organism that are associated with different hosts? Okay? It's common in chickens, it's common in cows, it's common in wild birds, etc. So, you know, largely, actually, we're working with Daniel Falouche and Xavier Didelot, who, I don't know if he's still here, but he was yesterday. You know, we started to think about applying GWAS to bacteria, okay? And we started with this Campy host model, right? So this is just a tree. It's highly structured, you can see. So straight away, there are differences with how we would use GWAS on bacteria compared to humans. And I suppose the key one to think about here is that if we're looking for adaptive elements that are associated with a phenotype, complex phenotype like host association, but any phenotype, if we're looking for adaptive elements, we don't really want to focus on elements that are just through common ancestry, okay? And it's quite difficult in a structured population to separate something that all these black ones have because they're adapted to wild birds from something that all these black ones have just because they share that common ancestor. Okay? So that's the key feature in making GWAS, applying GWAS to bacteria that's very different from applying it to humans. Okay, so how did we do it? Well, we could have looked at the black one compared to the blue one, okay? Or this cluster that's only found in chickens compared to this cluster that's only found in cattle. But we would have that problem of the kind of ancestrally inherited genetic elements. So what we did in this first study was we focused on these lineages, okay? So these you could consider generalists. There's blue and yellow in there, okay? And this one as well. There are isolates that we find in chickens and cows in there. And we postulate that because they cluster together but have divergent phenotypes, so they're from different hosts, there's been a relatively recent host transition. And we think that when that happens, because we're microbiologists, we think that bacteria adapt because a chicken gut is different to a cow gut. And we think that the adaptations that occur fastest are probably going to be, or the adaptations that increase in frequency in the population fastest are going to be the ones that are important for survival in the new host. Okay, so that's really fundamentally what our early GWAS model was based on. Okay, so this is just a methods slide. It's not terribly interesting, but of course you sequence lots of genomes. Our particular GWAS approach, and actually many of the ones that have come since, divide the genome into overlapping 30 base pair. We call them words, but you could call them features or chimeras or whatever you want. And then we sort these into two phenotype pools. In this case, the phenotype we're interested in is cow or chicken, right? Whether that isolate came from a cow or a chicken. And then here I'm just going to bang on a little bit about the population structure again, okay? So having done this simple association, we find words that are more common in one or another, and then we compare that to expectation based on the tree, okay? So if you just to say it again, if you think of these two blue ones, okay, if they've got this kind of increase in frequency of a particular word, that could be because it's an adaptation to living in a cow, or it could be because they co-inherited it. But if we also see it down here, okay, on a divergent lineage, then that's good evidence that it's associated with cattle adaptation. I'm not going to talk any more about that, but we can if you want later. Okay, so we did the GWAS. This is back in probably 2012. And this is a Manhattan plot. It doesn't look as jazzy as some of the human ones, but it's basically the same. And what we've done is we've found 9,000 words, okay, that are associated with one of our hosts. And then we've mapped them to a reference Campylobacter genome. And we find that they map to 99 words. I haven't written that on the slide. But many of them, 75% of all of our words map to a single region of the genome, okay, which is this part here, which is a cattle-associated hotspot, okay, in terms of sequence variation. We had a little look at the genes in that, and we just did some trees to get a sort of simple picture of how the trees of those genes might be different from those either side. And straight away you can see some difference, okay? They're not all stringy and branchy like these trees, but they have some sort of difference, okay? I didn't explain that in detail because it's explain in a bit more detail in this slide. And the thing to look at here, okay, so these are two of our generalist lineages that we mentioned. Yellow again is chicken and blue is cow. That's where the isolate came from. And these are the genes in that hotspot, okay, in the cattle-associated hotspot. So if you look at this curve, forget about the one on the right and just look at this one, you can see that in the chicken isolates, often the gene is missing. When there's no colour, that means the gene is absent. And also you can see that when present in cows, it's usually the same colour. That means it's the same allele, identical sequence, okay? So if you think about that, then you can start to think that maybe that could be evidence that these genes are conserved in isolates from cattle, because sometimes in the chickens they're not even there, and when they are there, they might have diverged a little bit. I'm not saying it's proof, just evidence. But really what we're thinking about is linking genotype to phenotype, the genetic basis of phenotypes that we think are important for whatever reason, whatever bug we're interested in this case, we sort of started to think about, okay, what's the function of these genes in this hotspot? Turns out many of them are to do with vitamin B5 biosynthesis. Well, I didn't know much about that before we did this study. In fact, I'd never really thought about it at all. Turns out, though, vitamin B5 is in all of the media that we use to grow Campylobacter in the lab. Okay, it's an essential nutrient for the growth of this organism. So then we were sort of thinking, well, why is that? You know, why is that? What's different about a cow and a chicken in terms of availability of B5? Well, one thing that is very different between cattle and chickens is what they eat. Chicken eat grain, which is very high in vitamin B5, and cattle eat grass mostly, I suppose, which has almost no vitamin B5. Okay? So when we think about that, what we can say then is that if we think this gene might be conserved in isolates from chicken, that kind of makes sense, because if you're a Campylobacter living in a cow, you would gain a big advantage from being able to produce your own vitamin B5. Okay? And yeah, okay, hey presto, in the lab it turns out that isolates from cattle can grow much better in a vitamin B5 depleted medium. Now I went through that rather fast because it's quite old now, and I think a lot of the people here have already actually seen me talk about this. So, but the point is that we got to an understanding of the genetic basis of a big difference between these strains without any a priori assumption, okay? And we made this story at the end that is feasible and can explain the genetic differences we see. All right, so since that time, there's lots of people, well, you know, lots of people have done lots of great things. You know, I particularly would encourage you to look at, there's a new pipeline from John Lees in the Sanger, and did anybody do my tutorial yesterday, the one that I did with John Lees? Excellent, excellent. And did it work? Well, it worked for me. No, don't say no. Anyway, so that's another GWAS pipeline that's sort of similar but different in certain ways, doesn't use a tree. And then Danny Wilson as well, I hope I've put a slide, has another one. So, but the point of this slide is just to show that, you know, it's a promising approach, and people really are starting to think that maybe this GWAS could be applied to lots of different things. Okay, so now I'm just going to talk really a little bit about how we're improving the GWAS and how we're trying to make this link between the kind of association of a SNP with a functional difference and sort of bridging that gap so we can actually make a kind of a really strong statement that our genetic variation is the cause of our phenotypic difference. Okay. So the first thing, so I'm starting with this, I'm going to talk about a few things, and then I'm going to come on to a couple of examples. So the first thing, you know, so when we're thinking about, well, I guess we're thinking about finding a phenotype in the noise. So typically we'll do a GWAS experiment on, I don't know, a few hundred bacterial genomes, and we'll get lots of hits for association. So we'll get lots of hits that make a difference in this group from this phenotype and this group from that one. And those GWAS hits, they are, of course, it contains the adaptive information that we're interested in, but it may also contain some of this kind of population structure, you know, the ancestral signal that we're trying to correct for by incorporating the tree. And it may also just have noise in there, you know. So how do we get rid of some of the noise so that we can really go for our adaptive signal? So one way, I'm going to whiz through these relatively fast because they're very simple, really. One way, initially we focused on these generalists, okay? So we're trying to reduce the effect of population structure. Well, in some of our recent work now, this is mostly stuff from E. coli, I think, for the next few slides. What we do now is we pair the isolates. So we pair isolates with very closely related genetically, but with a divergent phenotype. So in this case, it's E. coli in the environment and E. coli from animals, okay? And we can, and by doing that, we're trying to correct and remove as much of the ancestral signal as we can. Another thing in the early GWAS that we did was, if you remember the Manhattan plot, we mapped the hits to a reference genome. And of course, as you all know, your reference gene, there's loads of genes in your isolate collection that aren't in your reference genome, okay? So all of our GWAS work now, we use a pan-genome. So we basically take all of our isolates, identify all of the genes that are in them, all the open reading frames, and create a reference pan-genome, and then we map our GWAS hits to that, okay? So you can identify genes that aren't necessarily, that we might have missed in previous analysis. And just to sort of get an idea of how many that would be, for E. coli, our reference pan-genome for 246 strains was about 16,000 genes. When an individual one would have about 4,000 or 4.5 or whatever it is for E. coli. And then a third way of improving it, you know, is to do replicates. I mean, I'm a microbiologist, and I guess originally I was anyway, and you know, if I wanted to really test something, I just do lots and lots of replicates. Well, I guess the same is true in statistical genetics. So you can do replicate GWAS, which we now do, and this is on E. coli again. And you look for, you know, words that, lots of words in that one, lots of words in that one. You know, how many words are in both? You know, what's the overlapping signal in different data sets? Okay, and by doing all of this, you know, one hopes that you go from this very kind of difficult to interpret plot with lots and lots of associated words to something that's a bit more manageable. Okay? Right, so all of that is great. If you cast your mind back now to the figure I showed where we were talking about diet of chicken and cattle. Okay, so we found a signal in genes, and we found in the lab that we could explain it, at least, you know, theoretically, on the basis of this diet, okay? Because in the lab, we could show that they would grow better in a vitamin B5 depleted medium. But actually, there's a gap between there. You know, you need, this is kind of association, it's kind of a just so story. But really, we want a direct link to remove the faith, to return to the religion analogy. We don't want to think, we don't want to believe, we want to prove. Okay, so let's start by thinking about mutagenesis studies that we did on Campylobacter. A big thing is, how does it survive in the food chain? We know it's found in lots of farm animals, it's certainly in chickens, it's strongly associated with chickens. But what we noticed, okay, if we sequence lots and lots of isolates from different places, from farm chickens, from carcass meat in an abattoir or at retail, and then from clinical cases, okay, we find that some strains some increase in frequency as a proportion of the population through processing, and others decrease, okay? And it's sort of obvious from the kind of up and down lines on these graphs. So that gives us the idea that lineages might have different capacity to survive through the food chain. And if we can understand, you know, what it is that's making this organism strong enough to get through the food chain and get into humans, then maybe we can make some sort of intervention. So that's this kind of idea behind this study. So, you know, here's just a cartoon, but we're looking for enrichment of red elements, okay? That's basically what we're doing here. And the red elements are the ones that we think are associated with survival and virulence and other things, but, you know, they increase through processing. So we did our kind of standard GWAS, and we found our hotspots, you know, that were associated with survival in this case. In fact, we've worked a little bit more on it now, and we can identify the SNP that's responsible for our GWAS signal, our word association hit. And we can start to even think about, you know, haploblocks that might be associated with survival. So this is just showing the linkage disequilibrium decay, and we can see that our associated SNPs cluster together more than we would expect on the genome. But this is the bit that I wanted to talk about. Okay, so For those of you that know about Campylobacter, you'll know it's a microaerophilic organism. Sometimes it's hard to grow in a lab, and that's because it doesn't like oxygen, and it's not thought to survive well outside of the host. So it's no surprise that many of the genes that were associated with our survival through processing are to do with oxygen sensing and tolerance, okay? And this is kind of where we got to with the last Campy GWAS. We found genes that we could explain the significance. And also, we then went on to show that clinical Campylobacter strains actually in the lab were better able to grow at elevated oxygen, approximating atmospheric oxygen. Okay, but what I want is to fill in this little gap. I should have made it really wide, really. But which of these genes is responsible for this functional difference in the isolates? And what we did in this study was to try to specifically test the function of genes using knockouts. So it's sort of returning to the kind of microbiological background. So one in particular, I mean, I spoke to people that know more about this than me, but one in particular that showed a big signal was this NUK gene, which is associated with transition from anaerobic to aerobic conditions and the stresses involved in that. So we knocked this out and some others that are to do with NADH activity, and we found that the knockout mutants for NUK were much better able to grow in an oxygen-rich environment. Okay, so the point of that is that we're starting, unlike in human genomics, where we can't really mess with a human. You can do an association, but you can't then say, okay, well, we'll knock out that gene and see if, you get, anyway, whatever. You can imagine there are ethical reasons why you can't do anything like that. But in bacteria, we can actually make that link between the functional difference. Okay, so the final example is some work that we're doing on Staphylococci. And again, what we're trying to do is pick apart these complex phenotypes. In this case, it's virulence. But, virulence is a complicated thing involving lots of different factors. So, those of you who don't know what Staph Epidermidis is, well, it's on our skin. I'm sure you can guess that from the name. But it's a really big problem in hospitals. It's associated with infections of indwelling devices or catheters. It causes lots of bacteremia. In my local hospital in Swansea, anyway, it was probably as common as Staph aureus as a problem. It's a big thing. And this little schematic, this little cartoon, whatever you'd call it, sort of just illustrates the question that we're asking. Is this organism an accidental pathogen? Are all of the ones that cause disease, are all strains able to cause disease? Okay, so is it just that whether or not you get disease is just a function of whether or not you get a bacterium in your blood? Or are the pathogen ones, a subpopulation of the ones on the skin, that have this propensity, this capability to cause disease? Or is there some sort of overlap? Okay, so that's the question we were addressing here. So again, sequence lots of isolates from lots of different sources and do the GWAS. Okay, so this is presented slightly differently, but it's just kind of like a circular Manhattan plot. This is the chromosome of the reference strain. These are the plasmid genes, and these are the pan-genome ones from our kind of our reference pan-genome for Staph Epidermidis. And you can see again lots of hits, which is a challenge. So in the case of Staph Epi, what we did is we thought, well, okay, what is virulence in the context of an infection with this organism? It's lots of things, and we can start to sort of think about what they are in the lab. So there's toxicity, and there's antibiotic resistance, that might be associated with disease, and then there's host factors, the immune response. So you can't really read these. I probably should have made the writing a bit bigger, but basically these are the results of laboratory phenotype assays where we test these virulence-associated phenotypes. So the top two are IL-8 production from skin cells and blood cells that have been grown with Staph Epidermidis, different strains of Staph Epidermidis, okay? And you can see, the obvious thing to see is that there's very little immune response in the blood and quite a lot, on the skin, where it's commensal, and quite a lot in the blood. And then we also did toxicity assay, and we looked for differences in antibiotic resistance. But I mean, you know, so some of it's obvious, there's a difference there, but some of it's less obvious. But what we were able to do then is to use this data to screen our original GWAS hits, and that's what this funny little thing, you know, that I did on a train. I probably should try to do it a little bit better. But the point is, we do an original GWAS, okay, between our pathogen strains and our commensal strains, and then we get a lot of GWAS hits of associated words, but then we can screen those by a secondary phenotype. In this case, it's these things that we did in the lab, and we can see how many of our pathogenicity associated words can be explained by a phenotype that we observe in the lab. Okay? And this again is something that we just simply can't do for humans, you know, but this is a way that we can use our GWAS information with other things that we can do in microbiology to try and understand it. And so by doing this for every word, we can calculate how many, you know, we can give an actual score. I mean, that should be chi-squared, of course. I've said this before, but I just keep showing it because there's only 4 things. But some of our phenotypes are, you know, are continuous. So we use R-squared. And by working out how many of our GWAS hits can be explained by our phenotypes and giving it an R-squared, the probability, an R-squared value, we can work out, I don't know, well, we can start to screen through them and see if there's any way we can explain why that hit is there. So we've done this. The bands, if you just look at these associated words and the bands coming out, you can see that the biggest hits here are these kind of purple ones. This is where the R-squared is really high. And thankfully, when we did it on methicillin resistance, we find MEC-A and various other components of the SSC MEC element. So it works, you know, and that's quite a nice thing to do. Other phenotypes that we can test in the lab are a little bit more confusing and maybe we know less about. So we don't, I mean, certainly I don't know much about the genes we would expect to trigger a host immune response. And similarly, toxicity is quite complicated. But, you know, there's clues in here. I think somebody told me our AGRC was associated with toxicity. So, you know, we're starting to tease apart what we find within our GWAS work. And I think that's it. I think I'm going to end on that. But, you know, so really I suppose, you know, what I'm trying to get across here is that I think, yes, it's a promising technique. Yes, it's great. It's something we can do. We can use Climb for it. And I'm sure it's going to be quite a big thing in microbial genomics looking forward. But I suppose I want to try and get people to think about how we can use that data, not only from the point of view of statistical genetics, but think about how we can integrate that into the work that we actually do in our labs to really make this causal link between genetic variation and phenotypic variation in our organisms. So this is my most important slide. Thank you very much to all of these people. And of course, Daniel and Xavier, who were involved in all the early GWAS stuff with me. And John Lees is the chap that wrote the tutorial with me. I don't know who helped most, the other person there. Probably John did most of the work on that. But anyway, that was the GWAS tutorial that's on Climb, and I would encourage you to have a go if you're interested in this sort of thing. Thank you.